Thank you for introduction and thank you for invitation to speak. It's a great workshop, very interdisciplinary. So I disclose advisory role and IP belonging to NYU. This is a physics talk, and the physicist usually starts from length scales. So, you know, this is the molecular level where nanometers, where spins live. That's where we get our signal. This is our millimeter level resolution. Clearly, there are a lot in between. What exactly? This is kind of the intermediate scale. A physicist would call it a mesoscale because it's in between. In our MRI field, it's typically called tissue microstructure scale because the scale is roughly microns or fractions of microns, maybe tenths of microns. And that's the scale of cells or organelles. That's where all interesting physiology happens, pathology develops, and that's basically what makes tissues functionally different from each other and different from some kind of suspension of just proteins in water. And we want to access the scale, but we will really never get to this with brute force resolution. It's a fundamental problem. You cannot buy yourself out of it. So you need biophysical modeling. And what can give you this length scale that can access this cellular level? The process is diffusion. It was already described a bit by Susie in her talk. Let's remember, if you remember one number from this talk, it's this. Diffusion coefficient of water in tissues is roughly of the order of a micrometer squared per millisecond. So it's 1 in the units of microns in milliseconds. It's a very convenient number because we usually measure time in milliseconds in our scanner. And so, you know, the typical displacement is the square root of dt. And that basically converts your time to lengths. And so you get, you know, microns or tens of microns. And that is very fortunate, the length scale that is commensurate with cellular level. And that's, of course, 1 or 2 orders of magnitude below nodular resolution. So this is great news, but of course it's difficult to interpret the results. And just to give you an idea of the difficulty, let's assume very cartoonish model of the tissue. You know, there are these impermeable cells, and we only talk about in extracellular space. So spins perform random walk in between. What do we actually measure? So we apply a negative narrow pulse at time equals to 0 of the gradient, a negative gradient pulse. That means that you get each spin gets the phase proportionate to its position. And then after some time, which is called diffusion time, we apply the opposite refocusing pulse. And if the spin hasn't moved, you'll just cancel it. But of course it has moved. And each spin moves over different displacement. And this displacement, of course, is a probabilistic event. So they're weighted by this local diffusion propagator, the probability density of going from our point R0 to somewhere, to another point. And of course, because our resolution is the whole voxel, we necessarily average this quantity over initial and final points. So therefore, what we actually measure is a Fourier transform of this so-called diffusion propagator that is ensemble averaged over the whole all spins in the voxel. So essentially, all local propagators, they know a lot about microenvironment here versus here versus here, but you average them all together. And that is what you can access. And this is still great to have it. It's better than nothing, but you can imagine just amount of massive information loss, right? But because of this massive averaging. And to give you an idea, you know, the size of this, in the scale of the sizes of the cells, then the voxel will be bigger than a city block, right? So it's a massive averaging. So what we want is to see what information is not lost and to basically say something based on this about the geometry on the left. Okay, so we can take our signal and we can do 2 different conceptual things with it. We can either represent it in some basis. We can do a Taylor expansion, and the lowest order in this diffusion, the Q vector, Q up to Q squared gives you the diffusion tensor, and the next order gives you kurtosis tensor. And maybe some of you heard these words. Or you can take any other basis. You do not really understand what these coefficients necessarily mean, but that can already give you useful so-called quantitative biomarkers. But that just gives you sensitivity. If you actually want to learn about microstructure, you have to draw these cartoons. You have to model. Therefore, you have to represent the structure in some kind of geometric cartoons that are sufficiently parsimonious, but give you an idea what's happening. And that's really the essence of modeling. Model is part art, part science. It's like drawing sketches. Obviously, sketch number 2 is a better model than sketch number 1. And so that's what we do. We sketch microstructure and try to validate our sketches. And the challenge is what to include and what to leave out, because we want to have parsimonious pictures. So what can help us make these pictures parsimonious? That is the concept of coarse graining. So let's suppose we have a realistic axon, this axon segmented from EM electron microscopy, and we run non-color simulations within it. And so you can already kind of see that spins originated within a certain diffusion lengths from each other see more or less the same environment. So they do not really see something different. Therefore, if you have a particular diffusion time, you have a particular diffusion length scale corresponding to diffusion time, and you effectively homogenize the structure over this length scale. And the longer you wait, the more you essentially apply kind of Gaussian filter to this picture, the more uniform this structure becomes, because all spins more or less see the same amount of restrictions, so they're not different from each other. And so eventually you can see that axons become effectively a featureless stick. So characterized basically by only one parameter, diffusion constant alone. So diffusion is a coarse graining process over an increasing diffusion length, where details smaller than the diffusion lengths are washed out, and the relevant tissue features that we can quantify necessarily depend on the time scale that we import. Okay. And so that allows us to simplify things if we want to. So for instance, if you go to clinical, which is sufficiently long diffusion time, we can actually assume an axon for all practical purposes, a straight and narrow stick that is characterized by only one parameter, diffusion constant alone it, and 0 diffusivity in tensor direction. And so then diffusion is Gaussian in X, meaning also Gaussian in Q, since we're doing the Fourier transform. And the diffusion signal is also, of course, highly directional. If this B is a combination of Q squared and times called B value, that's what you control on a scanner. The larger this B value is, which is basically the strength of your gradient, the stronger is attenuation. And of course, the biggest attenuation is along the axon where spins move quite far, and there is no attenuation transfers of the axis because spins don't move, therefore there is no phase imbalance. Okay, so far so good. So now let's do one calculation that we'll do ever in this talk. Let's assume that the axon points upwards, and we pretend that we measure diffusion along all possible directions. Take the same p value, the same q, but vary the direction and average the signal over this shell of all possible directions. Take a directional average. What you'll get, well, it's an integral of this kind, but it's really, you can basically take it by hand. You basically realize that when you have very strong diffusion weighting, all the axons that are, all the directions that are almost parallel to the axon will give you exponentially suppressed signal. So the only signal that doesn't get suppressed is from directions that are almost perpendicular to the axis, because that's where the spins don't really move, there is no dephasing. And so basically you have this pancake of directions that is proportional to the 1 square root of this number b times dA, and b is our d value, that's what we control. So basically the signal, the spherical mean of the signal gives you this very slow non-exponential decrease as a function of b value. And that is very unusual because typically, we assume that in a bottle of water, our decay is exponentially strong, e to the minus bd. And here we get 1 over square root of b. it's incredibly slow. It's much slower than the exponential because of this fraction of the axons that are almost perpendicular to your gradient. And so that actually, that functional form, very non-trivial functional form, is a good way to test the hypothesis of whether we see the axis. So this is, you can actually go on the scanner and test it. And one of the most convincing evidences was actually obtained so by our team at the Honeycomb scanner in Cardiff, where we went to up to 25,000 B-value in radiological units and normal units at 25. So basically, this is the spherical mean at the enormously strong diffusion attenuation. You would put any other organ, not brain, in a scanner with this attenuation, you will see perfect noise flow. And yet here, you see the signal. And you see that white matter lights up. It's where the axons are. You're not doing spectroscopy. This is not like NIA or some metabolite. This is water, but it's only inside those impermeable tubes. This is kind of a miracle. This is how the signal looks as a function of a 1 over square root of B value. This is how we validate models. We predict very specific functional form and go to the very extreme experimental regimes where we can test that prediction. And we actually see that it goes as 1 over square root of B. The values goes from right to left. Right? And you still have like 5% of the signal, which is totally detectable. Whereas if you had like diffusivity of the order of 1 at this attenuation of basically in water, you'll get like 10 parts per trillion. You'll have nothing. That scaling validates the picture of axon as a stick. And it also justifies spending all this money, as Susie was alluding to build this super high performance gradient only for the brain because of this. Okay? So that has far-reaching consequences. So let us now demonstrate you how we do modeling. So, okay, so we validated elementary unit of our, say, white matter model, the stick. And then we look at a particular voxel, and this is EM segmented axons in 3D. And you see that they kind of come in bundles of fascicles. And within each fascicle, they're pretty aligned. So you can say we have perfectly aligned axons with just diffusion coefficient and water fraction. And then there is a coherent diffusion tensor, actually symmetric, of water outside of this fascicle. And then maybe also you can throw in free water because of this edema or some partial volume CSF nearby. And that is basically a single fascicle. So it has this scalar parameters, but of course they can be oriented in any way you like because there are fibers that are cross and a fan and so on. So basically you have the signal that is a spherical convolution of this elementary multi-component kernel, the fascicle response, and orientation distribution function. And that is known now under the name standard model of diffusion in white matter that we introduced a few years ago. And that really unifies a number of works over the past, I don't know, 10, 15 years, which were basically kind of trying to get to this picture. And they're included in this concept. Basically, other acronyms that some of you may have heard, NODI, WMTI, fiberball, etc., are just constraints on this picture. And we've done quite a bit of work on the subject to validate and study fit degeneracies and whatnot. Of course, the beauty now is that such modeling gives you specificity to distinct pathological processes, say intraaxonal is axonal loss or bleeding, extraaxonal parameters give you inflammation, demyelination, edema, and so forth, right? So why did we call it a standard model? Well, first of all, standard doesn't mean exact, better not. There's always something beyond it. This is a Lagrangian of a standard model in elementary particle physics. The name was suggested by tongue-in-cheek analogies with the standard model and the particle physics. In both communities, it represents the consensus knowledge about the subject. Yet everybody really hopes that there is exciting physics beyond. Okay, so for over $13 billion, particle physics community determined one parameter of the scalar potential, the curvature at the minimum, which gives you the Higgs boson mass. Unfortunately, it didn't get anything else. I just want to point out that for a tiny fraction of the cost, a number of microstructure groups around the world are studying biophysics beyond our standard model. Fundamental physicists in the audience, if somebody is contemplating changing fields, your time is now. So we put quite a bit of effort in parameter estimation. So there is a GitHub code for that. If you want to play with it and apply it to your studies, we basically do factorization. So this convolution is a factorization in spherical harmonic basis. So you can form rotational invariants and separate kernel and from the ODF and map invariants to parameters. That's how we do it. It needs validation, of course, as any model. So look at poster 26 of Ricardo Coronado Lieja. This is rat, the MRI rat model of traumatic brain injury, and you can see that axonal water fraction does drop in the TBI relative to healthy, and interaxonal diffusivity drops and axonal dispersion changes. And because we also have three-dimensional electron microscopy, we could compare one-to-one parameters such as axonal volume fraction with MRI from EM histology, gravitational dispersion, and even interaxonal diffusivity, and basically see that the parameters relate with their histological counterparts. And the standard model does show the highest specificity. So taking the details of poster 96. We can apply it to stroke, and you see that diffusivity interaxonal drops and axonal water fraction increases because of axonal beading. We can study the dynamics of pruning, myelination, axonal growth, and development. And one can also build on top of this picture. For instance, we realized that intraaxonal, extraaxonal compartment doesn't have to have the same T2 relaxation times, and actually they don't. The T2 outside is shorter than T2 inside, presumably because this extraaxonal space is more crowded and you have faster frequency to be defaced by the myelin. We actually can measure that, and you can add also T2 star relaxation and phase and build more complete multimodal pictures. And basically, this is the way to develop multimodality organically from the knowledge of microstructure, because microstructure drives all MRI contrast, whether we like it or not. And so understanding microstructure allows us to reap maximal benefit of multimodality, combining diffusion, proximity, etc. So we can now optimize the protocols for translating this picture. Say this is a fixed T, 15 minutes. If you want also T2 relaxation times, then it's a bit longer. And the reproducibility that is achieved is about 5 to 10% voxel-wise scan rescan, which is pretty decent. It's basically as good as well-done DTI. The poster 27, Valentin Stepanov applies this model with variable T to multiple sclerosis, and you can see the relaxometry changes and diffusivity changes, and highlighting edema and increased cellularity in the axonal space. We also ask this question, are those shells even optimal? Do we need to sample every shell in Q space? So because we don't know the orientation dispersion. And the answer is actually not. So the spherical convolution picture allows us to actually do only one direction per d-value. So if you have time for 100 measurements, you can do 100 different d-values, diffusion times, and sample each one with a different direction, and then reconstruct it all together. It's like Q space under something, mimicking K space under something. And actually, we could also involve K and Q space collaborating with Dmitry. So this is massive potential savings in position time, wide range of protocol optimization. So this is Santiago poster number 24. So the model so far, I told you, the standard model, enhanced by other multimodal processes. But what about the coveted biophysics beyond standard model? Well, actually, remember the coarse graining picture I told you about. While diffusion time varies, the diffusion coefficient decreases down to the kind of long time limit. And this decrease is basically manifestation of this coarse graining by diffusion. And actually, there is a theory we developed some time ago that this approach to long time limit happens slowly as inverse square root of time. And you can see it in the Monte Carlo simulations It's called diffusion of a function of one square root of time, and you can see a nice straight line. And that is because axons have all this regular shape, but not. And so there is a poster by Ali, those are the poster number 25, where we actually derive the exact relations between these two coefficients to the geometry of axonal cross sections, to beat amplitude correlations, the positions, and whatnot. So technically, this is given by this Feynman diagram contribution to self-energy part of the propagator, and the details are in Ali's poster. And so we can now, in the theory, apply this tens of thousands of axons segmented from 3D EM and predict diffusion MRI signal changes in chromatic brain injury in seconds rather than months of simulation. So this one was good time dependence has been confirmed also by our group and others in Viva. And so that is basically physics beyond quantum model along axons. Now think about transverse. So again, look at the picture I showed you before. This is white matter, one was square root of D value, and this is gray matter. They look very different. And none of them are truly looking as a linear one was square root of B for the feature of X. So what actually is the difference? So you see this white matter curve is slightly concave, and the gray matter is always convex. And that's actually quite important. So this concavity is a sign of finite axonal diameter or radius. And that's what Susie was alluding to. And there's quite a bit of research that has been done here and at NGH about mapping the axonal diameters. And we actually collaborate on the effect of irregularity in the axonal shapes on biases and estimation of those diameters. But if now you go and look for the gray matter, this convexity has a completely different explanation. This is actually coming from exchange between intraaxonal or intraneurite, now it's sticks of dendrite, and so between dendrites and extracellular space. And that's actually always convex, and you can get exchange times the order of 10 to 30 milliseconds, which are kind of accessible with human scanners. And so with Ileana Zhelezko, we recently developed this neurite exchange imaging framework to map exchange. And it's actually exciting because a good half of this exchange actually happens through active potassium sodium channels. So that those are metabolically regulated. So it's exciting that maybe diffusion can give you real-time metabolic activity. And there are also nice problems about interplay between exchange and structural disorder. And we're studying this together with Zhang group here at NYU and Wisconsin at GH. So we developed processing pipelines, denoising methods, artifact removal, but each of that will probably occupy the separate talk like this. I'll just say that microstructure gives you this push to become better at everything you do because there is such a high bar of data quality because model fits are pretty difficult. And so it pushed us to develop denoising and artifact removal methods to understand artifacts and noise well to actually make our fit robust, and as well as develop all the pipelines for validation and translation. And this is, of course, a team effort. So Mariana already mentioned Els Firmans, who's also sitting there. So Els and I have started this MR Biophysics group in 2012, 11 years ago, and we kind of covered together kind of modeling algorithms and clinical translation and validation. There is a lot of overlap, and these are our superstar alumni and our group members, a number of whom are here presenting the posters. And we don't often wear identical hoodies, but when we do, this is how we look. And I would like to finish this talk by quoting one of my scientific heroes, the late Freeman Dyson, who, after a stellar career in physics, one of the most original theoretical physicists of the 20th century, became world's most qualified futurist. He would always try to contemplate what the future innovations could bring. And I think that since I2I is the place where we contemplate the future, maybe it would resonate with you. I'm expressing possibilities, things that could happen to a large extent, it's a question of how badly people want them. The purpose of thinking about the future, it's not to predict it, but to raise people's hopes. I gave you the possibilities. In this talk, I showed you what is possible to have. Some of these parameters are very well established by now. Some of them, as for instance, exchange, are very much in active development. The choice, the compromises that we make, what to go for, is a large extent the question how badly we want them. I think this phrase really captures our tension between how much we push fundamental advances into the clinic. But I think the last phrase is also key. I think we should never forget that we work with a miracle device, a device that can allow you to pick orders of magnitude below the nominal resolution. And let's just hope that meetings like that can bring us together and we can eventually transform MRI scanner into a robust measurement apparatus for microstructure method. Thank you very much for your attention.
Thank you, Dmitry, for an outstanding talk. I think you answered in minutes, but maybe we can take one question.
It's Dmitry, it's very interesting. And you showed the way these two parameters, d infinity, and you said that it measures basically the level of Bouton type variations. I'm wondering, in white matter, kind of classical white matter bundles, there shouldn't be any Boutons kind of on the track itself. Is that correct, or am I missing something?
So it has been somehow concept that axons are kind of uniform, but they're not. And our group's efforts in 3D-EM and other groups just over the past few years show that normal axons, okay, fine, you have to fix them, and so it's not truly normal, okay, they're not live, but these are not diseased axons. These are 3D-EM, expression slice EM segmented axons.
Well, I guess, are they close to gray matter, or are they
No,
of
these
in
are
the middle
major tracks like corpus callosum.
track?
Yes.
Okay.
So
what
this,
do they connect
so this axonal beadings that are usually they're called, the distance between them is maybe 3-4 microns. Their amplitude could be, it could be variation of 50% or so. They're pervasive. They're everywhere. Go to Ali's poster. He'll show you beautiful.
Very interesting.
3D
Thank you.
And their geometry changes in disease. For instance, in stroke, they become more pronounced. But they're there originally already. And that's why this diffusion is never truly Gaussian. So we observe time-dependent diffusion along axonal tracts, even in normal volunteers, if you actually know where to.
Interesting. Thanks.
Okay, let's check again, Dmitry.